Well, good evening to everyone. I mean, Neha, good evening, Rahul, Khushbu, Mayank, Arsh, yes, Akash, Rahul, Ashok, good evening, guys, good evening. Welcome to the class, everyone. Live session with Collegeclue today, that is on molecular biology. Okay, today we will discuss. Well, good evening to everyone. Yes. Sonu Chaudhary, good evening. Rimsha, Shakir, yes. Very good, guys. Very good. Very good, Rahul. Very good. I'm very much fine, Rahul. Just waiting for another 5 minutes and then we'll start with the class. Just waiting for another 5 minutes and then I'll start with the class. Vipin, Shefali, Rahul. Yes, I'm very much fine. Yes. Yes, Nafas, welcome to the class. Today we'll start with the structure and organization of DNA. Okay, that is a very important topic in molecular biology to understand the structure of DNA. Vijay, good evening. Good evening, guys. Yes, good evening, everyone. Yes. Yes, just waiting. Rohit, Ankit, Kapil, welcome to the class. Yes, just for 2 minutes I'll wait and then I'll start with the class. Yes. Okay. Yes. Danish, very good, guys. I mean, I'm truly happy that you all are over here. Yes. Yes, I'll just start with in another 1 minute. Yeah, it's like 2 minutes are left, so I'm just waiting for everyone to join the class. Yeah. Hmm. So we'll just start within another 1 minute. Yes. Welcome everyone. Okay. Okay. Welcome Dushal. Yes. Welcome guys. Yes. Paramjeet, Arvind, very good. Sumit, Rahul, very good. Ashok, very good guys. I'm truly happy that you all are present. Yeah. Just a moment, I'm just working with the settings, guys. Yes. Okay. Okay. Gaurav Chaudhary. Yes. Very good, Neha. Very good. Good evening. Good evening, everyone. And now we'll start with the class. Yes. Dimple, good evening. Good evening, Neo. Good evening, everyone. Yeah. I'm quite happy that everyone has joined as of now. And now we'll start with the class. Yeah. Yes. Good evening. Good evening, guys. So let's start with another session in molecular biology where I will be discussing about the structure and organization of DNA. Okay, like you all know me, I'm Dr. Abhijeet Kumar. So my Instagram ID and Telegram group, you can like avail the benefits of the questions that I post on my Telegram account, on my Instagram account. Okay. Yes, good evening everyone. I mean, comments are just flaunting away. I mean, many of you have joined the class, so I'm quite happy about that. Chaudhary, Charan, very good. Very good, guys. I'm truly happy that you all have joined today. Yes. So now we'll start with the structure of the DNA. So put the heading as first. We'll just write about some general points about DNA. So put the heading as general points regarding DNA. General points regarding DNA. So yes, I hope audio and visuals are clear to each and everyone. Just give me a thumbs up, guys, in the chat box that audio and visuals are clear to each and everyone. Yes. So now we'll start with the general points. So the very first thing that I want to tell you about the discovery of the DNA. About the discovery of the DNA. Remember that we have a very yes. Yes. So yes, now we'll start, and I want every one of you to take out your notebook and start writing the notes along with me. Okay, I'm asking every one of you to take out your notebooks and just start writing the notes along with me. Yes, Sachin, Rahul, very good, very good. Dimple, very good, guys. Yes. So let's just start with the discovery of the DNA. Guys, remember, there is a lady that is called as the dark lady of the DNA. That is called as the dark lady of the DNA. And remember that dark lady of DNA, it is called as, she's the one, that is the Rosalind Franklin. Now guys, why she's called as the dark lady of the DNA? Remember guys that this is the one who is actually behind the discovery of the DNA. She was the one who actually first recognized the DNA, okay, and ultimately she couldn't publish it. That's why she's called as the dark lady of the DNA. All the work, all the findings were done by the Rosalind Franklin, but she was unable to publish it in the newspaper at that particular time, and that's why she didn't got recognized for this particular thing. And that's why, so now actually the structure of DNA, the structure of DNA is called as Watson and Crick model of DNA. Watson and Crick model of DNA. Now guys, Watson and Crick model of DNA, I have listened to a number of stories that tell that these were the people who were also working behind the discovery of the DNA, who were actually working on this particular project. But they actually got the papers, the pictures from the laboratory of Rosalind Franklin, and they were the one who actually first published the structure of DNA in the newspaper. So that's why now what we study nowadays, it's the Watson and Crick model of DNA, but ultimately it was discovered by Rosalind Franklin. But ultimately Watson and Crick were like, they published it first, they got the fame, that's why. And remember that it was discovered in 1953, okay? It was discovered in 1953, and remember, for which Watson and Crick, for which Watson and Crick was awarded Nobel Prize in the year 1962. In the year 1962. Kanchandeep, welcome to the class. I hope everyone is writing the notes along with me, and that's I want everyone to retain in this live session throughout, because You will be mastering the structure of DNA and the organization of DNA in this particular class along with me, and that will be the basis for the replication and transcription that I will do along with you in the upcoming classes. So for the basics, try to master the basic structure of DNA along with me. Take a pen, take your paper, write the notes along with me. Yes, Kailash, very good, yes guys. Abhishek, Garv, Kailash, very good guys. So now guys, we have already discussed about the discovery of the DNA in general, what are the dark lady of DNA, okay? And ultimately, later on, Rosalind Franklin died due to cancer because she was so much exposed to the X-ray, okay? So much exposed to the exposure to the X-ray. She was working in laboratory continuously for a long number of hours in the like okay, so ultimately she got died due to cancer, okay. So yeah, so the X-ray actually lead to her death because it ultimately turned out to be cancer, and later on she died. So that's why she remains always hidden, and whatever I have just researched on Google, so that ultimately says that she was the one who actually discovered about the DNA, but unfortunately she was not able to publish it, and it was published later on by Watson and Crick. Okay, so whosoever like publishes it will get the Nobel Prize as well. So yeah, that was in general, that was about the history. Now guys, we will start with the silent features of the structure of DNA. Put the next heading as the silent features. Silent features of structure of DNA. Silent features of the structure of DNA. I would request the technician. Someone is using my name and the chat box. Kindly block that person. Yes. Guys, stop behaving like if you don't want to participate. Someone, it's up to you. Just please leave the session right away. I don't have time for all this shit. And really, it cannot be deleted or altered in the live video as well. So we'll not waste time on that particular issues. Guys, please ignore them and just continue on what we are trying to focus on the silent features of the normal structure of the DNA. Okay, so remember guys, in the silent features, you can see that the first one, the first silent feature, it has two. We have already discussed the basics. It has two poly deoxyribonucleotide, deoxyribonucleotide. We have already discussed the structure of the nucleotides, okay. Sugar, phosphate and base. You all know that a nucleotide is basically composed of three things. That is the sugar, phosphate and base. Sugar, phosphate and base. And remember that sugar and base will actually continue to form the nucleoside. Sugar and base will continue to form the nucleoside, and ultimately if we are adding phosphate group to the nucleoside, we will actually land up in the formation of a nucleotide. Will actually land up in the formation of a nucleotide. So you already have, I have already discussed the structure of nucleotide, okay. And remember that it has two poly-deoxyribonucleotide strands, and these are running in right-handed helix. So they are just running in right-handed helix, okay. So that is the first feature, okay, running in right-handed helix. Right-handed helix. And remember that, okay, it can be, if I just want to compare it, this particular structure with a spiral staircase, okay. So remember this particular structure, okay, can be compared with a spiral staircase. You एक सीढ़ी देखिए बिल्कुल स्पाइरल वाली, ओके, यू विल फाइंड इट इन हवेलीज में, वो सीढ़ी देखने को मिलती है ना, राउंड टाइप, ठीक है, स्पाइरल स्टेयरकेस जिसको बोलते हैं, सीढ़ियाँ which is in the form of a spiral. So it is actually compared to this particular structure. So remember, it can be compared to a spiral staircase, to a spiral staircase, okay, where the handrails, where the handrails मतलब द साइड पार्ट जिसको आप लोग पकड़ के ऊपर चढ़ते हो, सो द हैंड्रेल्स जहाँ पे आप हाथ रखकर ऊपर मूव करते हो, सो दैट आर द हैंड्रेल्स which are formed by handrails which are formed by sugar and phosphate, which are formed by sugar and phosphate. And remember, and the steps are जो सीढ़ियों के जो steps होते हैं 1 2 3 4 the steps, they are basically formed by the non-polar bases. They are formed by the non-polar bases. Okay, bases we already have discussed. Purines, pyrimidines already have we have discussed adenine, guanine, thymine, uracil, cytosine, all the structures in a very easy concept manner I have discussed in the previous videos. So that was the very first silent feature of the structure of DNA. It's in the right-handed helix, and that too in the form of a spiral staircase. I hope that all the students are writing the notes along with me. Just give me a thumbs up that you all are present and writing the notes along with me, guys. Yeah, you all are present and writing the notes along with me. Yes, very good guys, very good. Yes, Deva, Vijay, very good guys. Yes, I'm happy that you all are participating today. Very good guys. Yes guys, notes साथ साथ लिखने हैं और I will check your notes as well after the class. So yes, apart from that guys, if we come to the second feature of the structure of DNA, the second feature of the structure of DNA, that is the anti-parallel nature of the strands. Anti-parallel nature of the strands. You know that we have two strands, okay? So ultimately, one strand basically run in the direction of 3' to 5' direction, and remember the other strand runs in the opposite direction. The other strand runs in the opposite direction, that is from 5' to 3' direction. That is from 5' through 3' direction. Apart from that guys, remember that was the anti-parallel nature of the strands. Okay, both the strands of the DNA are basically running opposite. Okay, like one from 3' to 5' direction and one from 5' to 3' direction. Okay, पहला वाला तो सीढ़ियों वाला कंसेप्ट था, जो सीढ़ियाँ होती है इट्स जस्ट लाइक दैट, और जो स्टेयर केसेस होते हैं दैट आर फॉर्म बाय द नॉन पोलर बेसिस. जो सीढ़ियाँ होती है, जो ये स्टेयर्स होते हैं दैट इज द बेसिस, और बाकी ये जो हैंड रेल्स होते हैं जहाँ पे आप लोग हाथ रखकर सीढ़ियाँ चढ़ते हो, दैट इज बेसिकली फॉर्म बाय द शुगर एंड फॉस्फेट. दैट इज बेसिकली फॉर्म बाय द शुगर एंड फॉस्फेट. Yes, very good guys. I hope everyone is able to like revise all the concepts that you have already studied in plus 2. So we'll just try to start from the basic and move to some more complex parts in a very easier manner. We'll try to discuss it. So remember that the third point, that the base pairs, okay, joins the two strands horizontally. Remember, horizontally by hydrogen bonds. आपको पता है कि if suppose this is the part of the DNA, if suppose this is the I'll just try to draw it nicely so that you are able to understand it. Yeah, if suppose this is one strand of the DNA and we have another strand of DNA, okay, we have another strand of DNA like this, and you guys can easily appreciate कि sir we have the bases like adenine, will actually pair with thymine of the other strand with two hydrogen bonds. Okay, so they are connected horizontally, ये horizontal है, और they are connected horizontally by the hydrogen bonds. एडेनिन जो है वो थाइमिन के साथ बाइंड करेगा, that too by the hydrogen bonds, that too by the hydrogen bonds. Okay, so remember, that is the hydrogen bonds which actually connects the two strands together, and that too in a horizontal fashion. Okay, so apart from that, here comes another rule just based on this, that is given by the Watson and Crick, that is given by Watson and Crick, and that is called Watson and Crick base pairing rule. Base pairing rule. And remember that Samik, I will first wish everyone should submit the notes which I am writing right now in the live class to my WhatsApp. And again, if I will find that majority of you have attended, I will share the PDF on my Telegram channel. Okay, on my Telegram channel, I will share the PDF. Just you have to be along with me, write the notes, and that is the okay, Watson and Crick base pairing rule. Now, what does this say? It says that the very first point, write down, guys, adenine pair with thymine by two hydrogen bonds, and remember that it is written as like this, that is A, that is adenine binds with thymine with two hydrogen bonds in a horizontal fashion. Okay, they are arranged horizontally. And the second base pairing rules, it says that it is the guanine which pairs with cytosine by three hydrogen bonds. And remember, guanine is pairing with cytosine by three hydrogen bonds. So that is the Watson and Crick base pairing rule. That is the Watson and Crick base pairing rule. I hope everyone is able to understand. Yes. So yes, just give me a thumbs up that, guys, you all are understanding right now over here. Yes, just give me a thumbs up. Everyone is present and understanding the concepts along with me. Yes. Jay, Vijay, very good, guys. Sayed, very good. Dimple, I mean, there's certain seconds of lap between I go live and you guys see on the screen. So yeah, it could be just certain seconds. Siddhi is also here today. Welcome, Siddhi. Ritik, girls, very good. Shoaib, Lakshita. I want every and each guy to respond in the live chat box, because that's the reason your presence would actually favor me to conduct the whole session again in future live. Yeah, absolutely, guys. I'm truly happy that you all are participating and writing the notes along with me. Okay. Now, guys, coming to another silent features of Watson and Crick model, the fifth one, and remember the fifth one, it is the number of purines is equal to number of pyrimidines. Number of purines is equal to number of pyrimidines. अब सर नंबर ऑफ प्यूरिन्स इज इक्वल टू नंबर ऑफ पिरिमिडीन्स, तो पहले सर प्यूरिन्स और पिरिमिडीन्स रिवाइज करा दो क्या होता है. तो प्यूरिन्स मैंने आपको बोला था, एक ट्रिक से याद रखना है, दैट ऑल गर्ल्स आर प्योर. ऑल गर्ल्स आर प्योर. प्योर मतलब प्यूरिन्स हो गया. ओके, ऑल से क्या हो गया, एडेनिन. एंड रिमेंबर दैट G stands for guanine. Okay, all girls are pure, so adenine, guanine. Okay, so remember that is the purines. And pyrimidines, basically you can remember that it is cytosine, uracil and thymine. Cytosine, uracil and thymine. Just remember by a mnemonic cut. Remember by a mnemonic cut. And you all know that uh, you if you forget it also, just guys remember कि pyrimidine में Y आता है, cytosine में भी Y है, thymine में भी Y है और uracil तो RNA में present होता है. So ultimately, and remember that pyrimidines are cut from purines. Okay, प्यूरिन्स का स्ट्रक्चर आप लोगों ने देखा था और मैंने बताया था कि pyrimidines हम प्यूरिन से ही निकालते हैं. So ultimately pyrimidines को निमोनिक आप लोगों को कट से याद रखना है. Yeah, that is the cut mnemonic that you have to remember. Now what does this rule says? So number of purines. So remember that A plus G, that is the adenine and the guanine, which are purines, will be equal to cytosine plus thymine, and that particular rule is none other than your favorite rule, that is the Chargaff's rule. That is the Chargaff's rule. And a question in NEET PGI 2013 came in the exam, and that question you guys will now do it and tell me the answer. That is the Chargaff's rule that you all have to like keep in mind, that the number of purines will always be equal to the number of pyrimidines. Yes, that is the Chargaff's rule. Yes, very good. Ashok, Kailash, very good. Very good. Very good, guys. So now guys, coming on to the question. Now the question says that a double-stranded RNA genome is isolated from a virus in the stool of a child with gastroenteritis. Now I'm discussing the clinical essential, guys. How clinical questions will be formed from basic molecular biology. How clinical questions will be formed from basic molecular biology, and that is the most potent question for next exam. That is the most potent question for next exam. पीजीआई एग्जाम में तो डायरेक्ट पूछा था, बट आजकल पैटर्न क्या हो गया क्लिनिकल क्वेश्चंस का. सो आई हैव ट्राइड टू फॉर्म दिस क्वेश्चन इन अ फॉर्म ऑफ अ क्लिनिकल सिनेरियो, एंड नाउ यू गाइस आर गोइंग टू टेल मी कि सर ये तो गैस्ट्रोएंटराइटिस है, एंड सी व्हाट इट इज saying. Remember, a double-stranded RNA genome isolated from a virus in the stool of a child with gastroenteritis was found to contain 15% uracil. What is the percentage of guanine in the genome? What is the percentage of guanine in the genome? Yes, 35, 35, 35. Guys, very good. Very good, guys. I mean, I am totally impressed. Okay. Now, guys, a certain trick. You all know that sir, number of purines will be equal to number of pyrimidines. Number of pyrimidines. Yes, that is the basic rule. Everyone is right. Sameek, Manisha, Vidhi, Pooja, Lakshita, Vijay, Jay, Ashok, Abdul, Garv, Aman, Gayatri, Shoaib. Very good. Very good, guys. I mean, I'm totally happy. But now I'll try to answer the question for those students who actually don't know. So that's very easy, guys. Very easy. Number of purines क्या होता है? You already know that कि sir A plus number of purines, that is adenine and guanine, will be equal to cytosine plus thymine. अब देखो यहाँ पर क्या गिवन है, 15% यूरासिल. ओके, तो ये थाइमिन की जगह यूरासिल भी हो सकता है. तो यूरासिल इज गिवन 15%. ओके, यूरासिल इज गिवन 15%. Yes, Sumit, I'm just answering your question. Yeah. Just wait, guys. I'm just answering your question. Yes. Wait. Okay. Now, can anyone tell me what will be the number of cytosine? What will be the percentage of cytosine? If like 15% uracil is given, what will be the cytosine over here? Okay, I'm waiting for your answers, guys. What will be the percentage of cytosine over here? Some are saying 35, some are saying 15. Okay. Okay, I'm waiting for the answers, guys. I'm waiting for your answers. I just wanted to know that how many of you have actually understood the question. Yeah. Hmm. I'm waiting for the answers. Yes, it will be like, okay. There's some connections going issues are there. Just a moment, guys. Yeah. Hmm, hmm, hmm. Okay. Yes. What will be the number of cytosine, guys? I'm waiting for your answers, guys. Meanwhile, I'll just check with the connections over here. Yeah. Hmm. Hmm, hmm, hmm. Okay, some are saying 15. Yeah. Kanchandeep, adenine is 15, guanine is 35, 35 cytosine. Okay. Okay, yes, Kanchandeep, you are somewhat right. Okay. Hmm. Yes. So I would just like to answer this question, guys. I like wanted you people to answer first, but yeah, many of you like got confused also. So remember that adenine plus guanine, okay, is equal to cytosine plus thymine. Remember that it is both 50% and it is also 50%. That's why these two things will be equal to each other. That two things will be equal to each other. Okay, guys. So remember that suppose now since, okay, okay, remember that, yeah, uracil basically binds with thymine. Oh, sorry, uracil basically binds with adenine. And remember that if uracil is 15%, what will be the adenine, guys, over here? Yes, if uracil is 15%, then adenine will also be 15%. Okay, adenine will also be 15%. And you guys know that since adenine plus guanine is equal to 50%, then what will be guanine now? It is 15 plus G, that is equal to 50. So guanine will be 35%, guys. Guanine will be 35%. Okay. Guanine will be 35%. Alternatively, guys, yes. Alternatively, guys, if you just want to see ki uracil is equal to adenine is equal to 55%. Okay. So ultimately, you can find that the guanine is like 35%. Okay, so that's the answer of this particular question. The answer will be C, guys. The answer will be C. So you all know that adenine basically binds with uracil. Okay, adenine basically binds with thymine in the DNA. If suppose thymine is not given, it is the uracil. So suppose uracil is 15%, so adenine will be the same. That is 15%. Okay, that will be 15%. So ultimately, if adenine is this, okay, it is like 15% and adenine plus guanine is equal to 50%. So what will be the guanine? It will be 35%. I hope everyone understood this particular question. I hope everyone understood this particular question. Just give me a thumbs up, guys, that you all like understood this. Yes. I would ask each and everyone to retain in the class, guys. I would ask each and everyone to retain in the class. Okay. Yeah. Yes. Okay. Okay. Now, guys, tell me one more thing. Can anyone tell me what will be U plus A? This is a question for you guys. If you have understood the concept, then tell me what is U plus A. I will be coming to the question. Like, tell me guys. Yeah, Shoaib Raja, I'm coming to the point also. Abnormal condition में हम लोग calculate ही नहीं करते. We don't calculate in any abnormal condition, guys. It's not applicable for any abnormal condition. That's why I'm coming to a percentage rule over here also. So now my question to you guys, what is uracil plus adenine? That's another question, guys, and you have to answer me. Okay? बच्चा 50 नहीं होगा ना, डीजे 50 नहीं होगा. अब खुद देखो यार, यूरासिल और एडेनिन बाइंड कर रहे हैं ना आपस में. एडेनिन इज इक्वल टू यूरासिल, एडेनिन बाइंड्स विद यूरासिल और थाइमिन. अगर अब सपोज क्वेश्चन में यूरासिल दिया हुआ है, तो अगर यूरासिल 15% है तो एडेनिन भी 15% होगा. तो अल्टीमेटली यूरासिल प्लस एडेनिन विल बी इक्वल टू हाउ मच? दैट विल बी 30%. दैट विल बी 30%. यस, एवरीवन इज राइट. नाउ गाइस, कैन एनीवन टेल मी if U plus A is equal to 30%, then tell me another question, guys. What will be C plus G? What will be C plus G? Yes, I'm waiting for the answers, guys. Yes, everyone is right who was like answered 30. तुम्हें पता है एडेनिन थाइमिन के साथ बाइंड करता है इन डीएनए, और अभी फिलहाल हम यूरासिल की बात करें, दैट्स इन द आरएनए. तो जितना यूरासिल है उतना ही एडेनिन होगा. ओके, इफ यूरासिल इज 15, एडेनिन भी 15 ही होगा. तो अल्टीमेटली टोटल यू प्लस ए करेंगे तो 30 हो जाएगा. Yes, Dimple Chaudhary, very good. Shakir, very good, guys. I hope everyone is participating. Yes, 70, very good. Garf, Pranjal, Neo, Siddhi, Kanchandeep, yes, very good, guys. So G plus C is equal to, G plus C will be equal to what, guys? It will be equal to 70%. Now guys, मैंने जो क्वेश्चन पूछा था आपसे इनडायरेक्टली, व्हाट इज द सी? नाउ आंसर मी कि सर साइटोसिन कितना होगा. ये क्वेश्चन में नहीं है, इट्स जस्ट अ क्वेश्चन फ्रॉम माय साइड. दैट इज द थर्ड क्वेश्चन यू हैव टू आस्क. what will be the percentage of cytosine over here, guys? What will be the percentage of cytosine over here? I'm waiting for your answers. Yes, Abdul, Ashok, Arshlan, Monishka, everyone is right. I mean, just seconds flaunt off हो रहे हैं. Yeah, I was just checking the connections that time. Yeah. Guys, don't leave the class. We are going to discuss each and everything in detail. Yeah, DJ, Kanchandeep, Uma, Siddhi, very good, guys. It is approximately, it will be 35%. It will be 35%. That was like all the questions that I have tried to cover over here. All the questions I have tried to cover over here. In any form it can ask you, and like it's the simplest way. You just have to put it like एडेनिन जो है सर वो थाइमिन के बराबर होगा या यूरासिल के बराबर होगा. ग्वानिन दिया हुआ है तो वो साइटोसिन के बराबर होगा. ठीक है, तो अल्टीमेटली यूरासिल दिया हुआ है, एडेनिन आ गया, और बेस पेयरिंग रूल में आप लोगों को पता है कि दोनों बराबर होते हैं. ठीक है, सो अल्टीमेटली यू हैव टू जस्ट कोरिलेट एंड आंसर द क्वेश्चन. दैट्स द सिंपलेस्ट वे यू गाइस कैन आंसर. यस, नफल बच्चे 70 नहीं होगा ना बच्चे, खुद देखो C प्लस G 70 है. अल्टीमेटली ग्वानिन और साइटोसिन भी इक्वल होगा ना. ओके, ग्वानिन इज इक्वल टू साइटोसिन. मैं अभी तो बोल रहा हूँ अगर C प्लस G 70 है तो ग्वानिन साइटोसिन के इक्वल है, तो मतलब ये तो दोनों बराबर होंगे. तो 70 का हाफ कितना होगा? अगर मैं G को x ले लूँ और C को भी x ले लूँ तो 2x बराबर 70, तो x बराबर कितना हो जाएगा? 70/2, दैट इज 35%. ओके, दैट इज 35%. आई होप यू गॉट माई पॉइंट. Okay guys, I mean, I'm totally happy with you all. Like you all are like participating. Yes, okay, chalo. Now guys, coming to some other question, and I want the answer of this particular question. That is the second question, that is the image based question. Nowadays in the NEET next exam, image based questions are being asked. Now I want the answer of this particular question. Okay, don't force me to guys, whosoever is like leaving the classes in between, it will be like a huge problem for them, because I'm discussing everything in detail and they have to be in the class. Yes, that is the question from my previous lecture, guys. Yes, so someone is saying nucleotide. Okay, What about else? Shakir says A. Is this a nucleotide, guys? No. No. चलो, वेरी गुड. ए है ये. विपिन, प्रांजल, शीनम, विपिन, तुम लोगों ने पिछला क्लास अटेंड नहीं किया. ए सी. ओ गॉड. दैट्स व्हाई गाइस आई ऑलवेज अर्ज यू ऑल टू अटेंड द लाइव सेशंस. तुम लोगों को यही नहीं पता चल रहा है कि सर क्या चल रहा है क्लास में. सो हाउ इट विल लाइक I don't understand how you guys are saying C also. Yeah, Arsh, you are absolutely right. Pranjal, you are also absolutely right. बच्चे सबसे पहले तो ये स्ट्रक्चर बताओ, किसका स्ट्रक्चर है ये? Which structure is this? छोड़ो, बेस का नाम छोड़ो, सिंपल बता दो ये प्यूरिन है कि पिरिमिडीन है. ये प्यूरिन है कि पिरिमिडीन है, ये तो पता चल रहा है कि ये बेस है. ठीक है, प्यूरिन है कि पिरिमिडीन है. जस्ट tell me guys. Yeah, it is the yes. I'm just asking. Yeah, deal. अब आंसर देने से क्या होगा? Like I'm totally like not happy about this anymore. Like you guys. हाँ बच्चे, ये प्यूरिन का स्ट्रक्चर है. मैंने तुम लोगों को बताया था प्यूरिन का स्ट्रक्चर. ठीक है, प्यूरिन का स्ट्रक्चर हम लोगों ने पढ़ा था कि सर यहाँ से स्टार्टिंग करते हैं नंबरिंग 1 2 3 4 5 6 7 8 9. जितने भी ऑड पोजिशंस हैं, ऑल ऑड पोजिशंस बेसिकली दे ऑक्यूपाई, मतलब ऑल ऑड पोजिशंस पे नाइट्रोजन प्रेजेंट होगा एक्सेप्ट 5. एक्सेप्ट 5 फिफ्थ प्लेस पे नहीं है. बाकी जितने ऑड पोजिशन है सब पे देख लो नाइट्रोजन प्रेजेंट है. और यस, कैन एनीवन टेल मी द नेम ऑफ द बॉन्ड विच इज प्रेजेंट ओवर हियर? आई एम मार्किंग एज बी. Can anyone tell me the name of the bond which is actually joining the base with the sugar? Base with the sugar. ये शुगर मॉलिक्यूल है और इस शुगर मॉलिक्यूल का नाम भी बता दो. ये शुगर राइबोस शुगर है या डीऑक्सी राइबोस शुगर है, ये भी बता दो मुझे. आई टोल्ड यू ये शुगर राइबोस है या डीऑक्सी राइबोस है, ये भी बता दो मुझे. ये मैंने पिछले लेक्चर में आप लोगों को बहुत क्लियरली बताया था. आई जस्ट वांटेड टू डिस्कस how like you guys are paying out attention or no, like DJ ये phosphodiester bond है. I mean I cannot believe this. Arsh, yes, very good. Arsh, Shakir, very good. Kanchandeep, very good. It is the beta N glycosidic bond. Beta N glycosidic bond that is present between the base and the sugar. The base and the sugar. Yes, it is ribose. Ribose में दोनों जगह पर थर्ड और सेकंड दोनों पोजीशंस पे OH ग्रुप प्रेजेंट होता है. डीऑक्सी राइबोस होता तो इट विल बी ओनली OH ग्रुप सिर्फ थर्ड पोजीशन पे प्रेजेंट होता. यस, और बेस और शुगर जब मिलता है तो दैट इज द फॉर्मेशन ऑफ अ न्यूक्लियोसाइड. न्यूक्लियोटाइड तब बनता बच्चे जब यहाँ पर मैं फॉस्फेट जोड़ देता. जब यहाँ पर मैं फॉस्फेट जोड़ देता, बट यहाँ फॉस्फेट अटैच्ड नहीं है तो ये न्यूक्लियोटाइड नहीं है, ये न्यूक्लियोसाइड होगा. दैट विल बी द न्यूक्लियोसाइड एंड दैट इज अगेन अ प्यूरिन न्यूक्लियोसाइड. दैट इज अगेन अ प्यूरिन न्यूक्लियोसाइड. ओके, सो द आंसर विल बी डी. आंसर विल बी डी. पिरिमिडीन में एक ही रिंग होती है ना बच्चे. पिरिमिडीन और प्यूरिन में फर्क ही यही होता है, प्यूरिन में 2 रिंग्स होती हैं और पिरिमिडीन में एक होती है. तो हमेशा मैं लिखवाता हूं ना अपनी क्लास में कि नो, pyrimidines are cut from purines. प्यूरिन्स में से एक रिंग काट दोगे तो पिरिमिडीन बन जाएगा. और कट तो तुम्हें पता ही है, साइटोसिन यूरासिल थाइमिन. साइटोसिन यूरासिल थाइमिन. यस गाइस, यस, साइटोसिन यूरासिल थाइमिन. साइटोसिन यूरासिल थाइमिन. Yes, very good guys. So I hope everyone understood that it's a purine nucleoside. बच्चे यहाँ कोई फॉस्फेट ग्रुप नहीं जोड़ा हुआ ना, तो अल्टीमेटली जब फॉस्फेट, शुगर और बेस तीनों मिलते हैं उसको न्यूक्लियोटाइड बोलते हैं. यहाँ पे तो कोई फॉस्फेट ग्रुप नहीं जोड़ा हुआ है. सो अल्टीमेटली दैट इज द लाइक yeah, that is you guys have to be very much clear about. Okay, that is you have to be very much clear about. So just give me a thumbs up that you all understood right now. Yeah, you all understood right now? Yeah, everyone is there participating in the lecture, I suppose. Hmm. Okay. Very good, guys. Very good. Very good, guys. Hmm. Sheenam, Lakshita, very good. Very good, Ashok. Yes, very good. Hmm. Sure. Anyways, we'll just move further. Okay, we'll just move further. I'm just reading out your comments. Like everyone is participating, and that's very, very good. Okay. Sure. Yes. So we'll just like move ahead now. Okay, we'll just move ahead now. Yes. Okay. Okay. Sure. Let's start with another feature. We have already discussed the purines and pyrimidines, that is the Chargaff's rule. We have already discussed the purines and pyrimidines, the Chargaff's rule. I hope that's automatically clear. Adenine plus guanine will be equal to cytosine plus thymine. Adenine plus guanine will be equal to cytosine plus thymine. Now guys, we will start with another rule, okay? The another silent feature of the DNA, guys, another silent feature of the DNA, and just try to remember it, guys. Yeah. Okay, so we'll just start with, yeah, my phone just got disconnected. I cannot see the charging part over here. Yeah, so yes, now moving on to the another part, guys. Yes. Okay, Anshul, very good, guys. Just now start with the next part. Okay, we are now going to study the major that in a structure of DNA we actually have the major grooves, okay, the major grooves and the minor grooves, the major grooves and the minor grooves. Okay, so remember the major grooves and the minor grooves, the major grooves and the minor grooves. Your voice is like coming. Yeah, the major grooves and the minor grooves in the DNA, in the DNA basically are the sites where proteins interact with DNA, where proteins interact with DNA. I'll just check the connection over here. Yeah. Yeah, I hope it's fine, guys. Now I hope it's fine. Yeah, I've just checked my connection. It's like working very good over here. I just don't understand. It's like okay. Yeah. Yeah, from my side it's okay, guys. From my side it's like working well. Just check your internet connections. Yeah, please just check your internet connections at your end, guys. Okay, at my sides like the connections are very well like working good. Yeah, so I don't like understood what has happened. Okay, so we have the major grooves and the minor grooves, guys. So we have the major and the minor grooves, guys. So apart from that, guys, now we'll start with the base stacking part. The next, that is the base stacking. अब इसमें एक डायग्राम में सब कुछ समझ आएगा. So just we'll try to draw the diagram and we'll try to understand the concept of base stacking now. We'll try to understand the concept of base stacking now. So remember guys, the base pairs. You already know that we have different base pairs, and remember they stack one above the other, one above the other by a vertical interaction, by a vertical interaction. And remember the vertical interaction between the base pairs, it is actually called the van der Waals forces. Base pairs, it is called the van der Waals forces. It is called the van der Waals forces. Now suppose guys, we are just going to try to draw a structure of the DNA over here, and like now I'm going to draw the another part of the DNA, the other strand of the DNA with another pen, that's 2 from 5 prime to 3 prime direction, and another strand that I've already drawn, it is from 3 prime to 5 prime direction, 3 prime to 5 prime direction, 3 prime to 5 prime direction. Okay, 3 prime to 5 prime direction, 3 prime to 5 prime direction. Yes, I hope everything is like going smooth now. Okay, yeah, everything is going smooth now. Yes, guys, truly participate in the class throughout, and that's what is required as of now. Okay, so guys, remember that we have 3 prime to 5 prime direction, that are the two anti-parallel strands of the DNA. Remember that अगर guys यहाँ पर adenine present है, now I am talking about the bases over here. If suppose adenine is there and thymine is on this particular strand, they both will connect to each other by hydrogen bonds. Okay, two hydrogen bonds are there. If suppose guanine is there, it will bind with cytosine of this particular strand by 3 hydrogen bonds. Okay, 3 hydrogen bonds, and over here we have the 2 hydrogen bonds. Over here we have the 2 hydrogen bonds. Now guys, try to see कि अब guanine के नीचे भी adenine के नीचे भी guanine present है and it's binding with cytosine. Okay, so now what is base stacking? Now these base pairs ये horizontally तो connect होते ही हैं hydrogen bond से. These are connecting horizontally with the hydrogen bonds guys. Okay, now what will happen? These will actually connect to each other vertically also. These will connect to each other vertically also. Adenine with guanine, thymine with cytosine vertically, and that is by the van der Waals forces. And that is by the van der Waals forces, and that is called as the base stacking. That is called as the base stacking. एक बेस पेयर के ऊपर दूसरा बेस पेयर, उसके ऊपर दूसरा बेस पेयर, उसके ऊपर दूसरा बेस पेयर. So they are actually interacted in vertical direction also. They are again interacted in vertical direction also. Yes. Guys, I like don't know why this is like happening as of now. I hope the connections are fine at my end. Like, no, my Wi-Fi is actually working very good over here. See guys, if something is there, just let me know. Like, okay guys, like I cannot like waste more time. Just try to understand guys that it is the van der Waals forces which is actually connecting the base pairs one above the other by vertical interaction. Adenine भी guanine के साथ connect करता है बच्चे, that's by the van der Waals force, and that is called as the base stacking. That is called as the base stacking. Yeah. So just try to concentrate, guys. Don't leave the class. I am urging everyone to kindly learn along with me, write along with me. Yes. Now guys, remember कि ये जो distance होता है thymine, अब suppose I'm just trying to draw a little big for you guys so that you can understand it more clearly. Remember, now suppose this is the strand of the DNA and this is the other strand of the DNA. Now guys, remember we have the base pairs over here, that is the adenine, which is binding with thymine with 2 hydrogen bonds, and guanine binding with cytosine with 3 hydrogen bonds. Now guys, try to see over here that we have between the adenine and guanine there is a force of attraction vertically. ऊपर से नीचे vertical attraction भी होता है बच्चे. It is not only the horizontal attraction, that is the 2 double bond, 3 double bonds. It is also the vertical interaction from like in a vertical direction, you can see, and that is the van der Waals forces. That is the van der Waals forces, and that is called as the base stacking. And that is called as the base stacking. Now guys, try to see कि इन दोनों बेस पेयर्स के बीच में वर्टिकल डिस्टेंस जो होता है, वर्टिकल डिस्टेंस बिटवीन दीज टू बेस पेयर्स, ओके, remember that it is around 3.4 Armstrong. It is around 3.4 Armstrong, or in nanometers, if I want to write it, I will write it as 0.34 nanometer. 0.34 nanometer. And remember guys, that yeah, this is the bond. So write down this point, the bond forming a van der Waals force. I will check the notes after the class, guys. Everyone has to submit the notes on my WhatsApp, and that's very much required. I want each and every one of to be in the class and write the notes. It's for you people, okay? It's for you people, okay? So just the bond forming distance of a van der Waals force, okay, is 3.4 Armstrong. Or if I want to write it in nanometer, it is 0.34 nanometer. It is 0.34 nanometer, guys. It is 0.34 nanometer. Apart from that, remember guys, in one turn of DNA, okay, if suppose in one turn, so this is actually one turn of DNA. From this point to this point, it is called as one turn of DNA. And write down, in one turn of DNA, basically there are 10 base pairs. There are 10 base pairs. In one turn of DNA, there are 10 base pairs. Okay, so remember कि यहाँ पर अभी तो मैंने आपको 2 दिखाए हैं, यहाँ पर 10 बेस पेयर्स होते हैं, यहाँ पर 10 बेस पेयर्स होते हैं, 1 2 3 4 5 6, एक टर्न ऑफ डीएनए में 10 बेस पेयर्स होते हैं, और ठीक है, एंड दैट इज द रीजन कि हाइट जो है वन टर्न की, आई एम नॉट टॉकिंग अबाउट द हाइट ऑफ वन टर्न, रिमेंबर द हाइट ऑफ वन टर्न, रिमेंबर इट इज 34 Armstrong or 3.4 nanometer. Okay, it is 34 Armstrong. Just हमें इसे 10 से मल्टीप्लाई करना है. हाइट ऑफ वन टर्न कैसे निकालेंगे सर? एक के बीच में डिस्टेंस तो 3.4 Armstrong है. आई विल मल्टीप्लाई इट विथ 10 बिकॉज 10 एक टर्न में 10 बेस पेयर्स है, तो इट विल बी 34 Armstrong और 3.4 नैनोमीटर. इसको जस्ट 10 से मल्टीप्लाई कर दो बिकॉज 10 बेस पेयर्स है एक टर्न में. ओके, एक टर्न में. ओके, so that is the like, yeah, in one turn you will get these 10 base pairs. Okay, guys, I simply want kindly ignore these type of people and focus on the content that I am giving you right now. Kanchandeep, Vidhi, just ignore this Sinchan and other like whosoever account is there. If the technician is watching, kindly block these people, please. I just want to block these people right now because they are ultimately disturbing my class. Yeah. So guys, remember that you already have done, that is height of one turn. It is also called as the pitch of the helix. It is also called as the pitch of the helix. हाइट ऑफ वन टर्न को हम लोग पिच, मतलब इस पूरे स्ट्रक्चर को हम लोग पिच ऑफ हेलिक्स भी बोलते हैं, पिच ऑफ हेलिक्स भी बोलते हैं कि सर वर्टिकल डिस्टेंस वंडरवॉल फोर्स के बीच में 3.4 आर्मस्ट्रॉन्ग है और एक हाइट में 10 बेस पेयर्स होते हैं, तो इसको 10 से मल्टीप्लाई कर देंगे तो हाइट ऑफ वन टर्न मिल जाएगा. दैट इज ऑल्सो कॉल्ड एज द पिच ऑफ द हेलिक्स एंड अल्टीमेटली इट इज 34 आर्मस्ट्रॉन्ग. दैट इज 34 आर्मस्ट्रॉन्ग. Okay, I hope guys you understood this particular part. I hope all the basic general structure of DNA is clear to each and everyone over there. I just want, yeah, everyone like has understood all the basic structure of the DNA and I have just shown in a very clear, crisp manner. Okay, I hope everything is clear. Yes. Okay. Very good, guys. Very good. Very good. I'm like very happy that you all are participating and like answering the trying to make the notes along with me, guys. That's very, very required. So all the silent features of DNA, all the 6 points, the major minor grooves, the Chargaff's rule, everything I have discussed along with the questions that can be asked in the upcoming next exam. Along with the questions that can be asked in the upcoming next exam, I have already discussed. Now guys, another topic that we will focus right now, it is the types of DNA. It will be the types of DNA, guys. Types of DNA. Types of DNA, guys. Yes, very good, guys. Very good. If you all are participating, I'm quite happy and I want like every one of you to like participate. Okay, I want every one of you to participate. Those students who are not participating, kindly ask them to join the class, to attend the lecture. It's very much required as of now, okay, to participate and to learn. Okay, whatever I'm just teaching you guys, it is for your benefit, guys. It's for your benefit. Yes. Now guys, we'll start with the types of DNA. And remember that there are 6 types of DNA. There are 6 types of DNA. And remember that it is A, B, C, D, E and Z. Now why I have written Z a little away from all these, because there is a major difference that A, B, C, D, E are all right-handed DNA. Okay, they are arranged in a right-handed fashion. Yes. So yes, it is a right-handed DNA. It is a right-handed DNA, whereas Z is basically a left-handed DNA. Z is basically a left-handed DNA. Left-handed DNA. Okay, it's arranged in. Now guys, I'll try to tabulate the differences between all the DNA. So just make a table, guys. Make a table, guys, and just try to, I will try to tabulate the differences between the DNA, that is A DNA, B DNA, which is like the most important ones, and the C DNA. The C DNA. Now guys, see, the very first point that how to differentiate between these 3 types of DNA, the very first parameter that I will take, it is the direction of turn. It is the direction, or not C, it is Z. It is Z. Okay, it is Z. Okay, so just write down it as Z. Okay, so yeah, C is not like very much important as of. I'll just mention some points afterwards. So yes, it is the direction of turn. Direction of turn. Yeah, so the direction of turn in case of A, the type of DNA, that is A type, and you can see it is a right-handed. It is a right-handed. B, it is also right-handed. B, it is also right-handed. And Z, it is the left-handed DNA. It is the left-handed DNA. It is the left-handed DNA. The other parameter that I will try to distinguish guys right now over here, it is on the basis of the number of base pair per turn. The number of base pairs per turn. मैंने अभी तो वैसे आपको per turn जो हमने एक पिच में पढ़ा था, that is the height. Okay, एक पिच में हमने 10 base pairs पढ़ा था, that was like in normal, in general. But A, B और Z में remember that in A type of DNA it is the 11 base pairs. It is the 11 base pairs. B, it is 10.5 base pairs. B, it is 10.5 base pairs. And Z, it is 12 base pairs. Z, it is 12 base pairs. Apart from that guys, remember that the base pair tilt, the base pair tilt corresponding to axis of helix, corresponding to axis of helix. So the base pair tilt at what angle it is actually corresponding to the axis of the helix. Okay, so how this tilt is basically reacting or corresponding to the axis of the helix. Okay, so yeah, it is around 20 degrees in type A, 90 degrees in type B, and 9 degrees in type type Z. Apart from that, one more parameter that I wanted to like use as a parameter to distinguish between the three types, it is the morphology guys. It is the morphology. And remember that A type of DNA, it is broad and short. The B type of DNA, it is elongated and thinner. And Z type of DNA, it is elongated and thin. Elongated and thin. So that is in general about three types of DNA. Okay, the three types of DNA and the basic differences between all that, and the basic differences between all these. Now I will try to talk about these DNAs like one by one. So write down the very first one, that is the A DNA guys. That is the A DNA. And remember that A DNA is found in a region. It is found in a region where there is low humidity, low degree of hydration, and high degree or high salt concentration. So that is about the ATNA. It will be found in a region where there is low humidity, low degree of hydration, and high salt concentration. Okay, automatically जितना पानी कम होगा, the concentration of salt will be more. Okay, so apart from that, if we talk about the BDNA, it is found in a region. Where there is, just opposite to this, high humidity, high degree of hydration, and low salt concentration, And remember, a question has been asked in the FMG exam earlier that BDNA, it is the most common type of DNA. So BDNA, it is the most common type of DNA. And remember, it is physiologically most stable DNA. And it is physiologically most stable DNA. it is physiologically most stable. These are two different questions and they have been asked in the exams. Okay, ultimately high degree of hydration, high amount of water concentration is there, so ultimately it will be the most stable part. Okay, it will be the most stable part of our body is like more type of water. Okay, so ultimately it is the, yeah, A and the B DNA, A and the B DNA, guys. So I hope everyone understood about the different types of the DNA, the all the basic silent features of the structure of DNA, the general points about the DNA, the general points about the DNA. Guys, like I wanted to continue the lecture, but yeah, I will be now willing to continue it tomorrow again live at the same time, guys, and I'll discuss the structure of the DNA. For today, just let me know if you guys have any issues regarding the general points, the silent features that we already have discussed with two different questions, okay, and yeah, with two different questions, yeah, and yeah, the types of the DNA, guys, the types of the DNA. I hope all of you understood the lecture very well today, and I hope everyone has prepared the notes. Yes, everyone has prepared the notes along with me. Yes, so I'm just waiting for your, yeah, yeah, responses, guys. Yes, I'll just continue the lecture tomorrow as well. Don't worry, I'll complete the topic. I'll just discuss the structure of the DNA as well, so equally don't worry. No Duke of Doom, Wonderworld forces are present over here also. It's not only for gases. सिर्फ अगर तुमने solid, liquid और gases में उधर 11th वाला concept उठा के मत बोलो. You are studying medical now. Okay, so yes, I hope everyone understood the lecture very well today, and everyone has like made the notes. Yeah, the timing will remain the same, guys. I'll share you the PDF as well. Meanwhile, you can follow me on my Instagram, like all the details are being displayed on the screen. For any particular queries, any particular doubts, you are most welcome to me. Daily, guys, be with me daily. One नहीं यार, अपोजिट साइड भी हो सकते हैं. दो अलग अलग न्यूक्लियोटाइड्स है. इट कैन बी अपोजिट साइड्स ऑल्सो. It can be opposite sides also, Ajay Kumar Yadav. Okay, it can be opposite sides. It's not compulsory. Okay, yeah, guys. I hope everyone understood. Yeah, it can be opposite sides. Any doubts, guys? Please write in the chat box. I will be happier to address them right now. As Ajay asked, I have already told that it is not always the same side. It can be like vice versa as well. Okay. Any guys, any particular doubt, just let me know. We'll have a session tomorrow also, I suppose. If I'll get time, I'll definitely take a session. And yes, a little bit tired today, but still I just promised that I will be taking the session, so that was like a promise and I just fulfilled it. Yeah, yes, I'll just see. Okay, guys, I hope all the points are just clear to everyone and all the questions that we have discussed and the types of the DNA along with the most important questions for the next exam. Yes, yes, good night guys, and thank you. Yeah, it is like 10.5 base pairs, nearly possible, guys. Okay, uh, yeah, nearly possible. Okay, it's just present on only one strand Danish. That's why bacha, not on the other part. It's just on one other part, guys. And those whosoever has attended my lecture with full sincerity, text me on the my WhatsApp that with the notes, guys. I will be happier to see them, and like it will be positive energy for me also to give you class for the next time, to give you a session next time as well. Thank you guys, God bless you everyone, and yes, I will be. बच्चे 10.5 है आप हाँ हाँ उसमें था, ठीक है. Danish, text me on WhatsApp. I'll explain this particular point. I already told, yeah, it's just present on one strand of DNA, not on the other. In that way, it's saying like that. Just text me on WhatsApp. I'll more happier to address you on call. Okay. हाँ बच्चे, हाँ लिखवा दिया है शोएब. नोट्स पलट के देखो अगर साथ में लिखा हुआ है तो, है ना? So yes, thank you everyone, and yeah. Yeah, good night, good night, good night, guys. Good night. Good night, guys. I'll just end the session.